I think we should go. Dipti?
Yeah, okay. So should we start now? As I can see that, yeah, the participants, they are joining. So should we wait for 1 minute or I can just go ahead?
Let's go ahead. Yeah.
They are still joining. Okay. Okay, so I welcome you all for 5th ETC Auditorium. And our today's speaker is Juan Antonio Vizcaino, who is currently a proteomics team leader at EMBL European Bioinformatics Institute in Cambridge, at United Kingdom. And he is mainly responsible for the development and the maintenance of the PRIDE archive, which is a world's leading repository for mass spectrometry-based proteomics data. And so he's also been the manager of Proteome Exchange Consortium from 2011. And he has contributed immensely in the development of proteomics standard data standard formats. He's also a co-chair of HuB PSI, which is a proteomics standard initiative community. So, and from a little of his background, that he did his PhD from University of Salamanca, Spain, in microbiology in 2005. And then he carried out his postdoctoral research for 1 year as a bioinformatics data analyst at University of Seville, Spain. And then from 2006 to 2008, he was a postdoc fellow at EMBL EBI. And then he joined as a project group coordinator from 2010 to 2015 at EBI. And now he's leading the proteomics group at EMBL EBI. So let's not wait for more. And I ask Juan Antonio to start his presentation.
Hello, Dipti. Thank you very nice for the kind introduction. I'm just going to share my slides. Can you see the slides? I think so, no? Hopefully
yes,
you should be able to see.
Yes, we can see. Yeah.
Okay, so first of all,
but it's not in the presentation mode. It's present mode. You can.
Okay, so let me try. One second. Yeah, 1 second. I will just stop sharing and I will share again. Okay. Apologies, I have many different many different screens and some screens, and it should be fine. Now?
No, it's fine. Yeah.
Now it's fine. Okay. So first of all, I would like to thank the Education and Training Committee from UPO, especially Tianan and Dipti, for having invited me to give this webinar. And I hope that the most important thing is that you find it useful. I'm happy to reply to any questions you may have after, or if not, contact me by email afterwards. Okay. So the title of my presentation is Open Data Practices in Proteomics: the Why, the How, and the What for. So first of all, I thought that it was good to include a couple of slides to give some background about where we are based. So we are working in the European Bioinformatics Institute, EMBL, which is based in the south of Cambridge. And it's really the world-leading source of public biomolecular data. Really, our vision is to benefit humankind by advancing scientific discovery and impact through bioinformatics. And I'm sure that most of you must have used at least one, or if not many, of the resources that are provided by the European Bioinformatics Institute. And there are many, I don't know, from Ensemble and Uniprot and Protein Data Bank and the EBA and the EGA, and so many resources that I'm sure you must have used. My team is responsible for PRIDE, the PRIDE database, like you will be listening to me later. Okay, so after this very short preamble, I'm going to give an overview of the talk. First, I'm going to talk about the why sharing proteomics data is a good thing. Then the how, and I will talk about protein exchange resources and the PRIDE database in particular, but I will also talk about other protein exchange resources. And the what for, I think maybe is the most important thing in the talk, is just to show you some examples of data reuse of public data sets to see what can be done with that data. And I will end just with one summary slide about future perspectives or important things for the future. Okay, so I hope that, as I said, you will find this interesting. So about the why. So this is the first question that I would like to answer in my talk. So why it is important to share proteomics data in the public domain? So as we all know, if we are working in the field, proteomics data can be very complex, and in its interpretation is often difficult and in some cases controversial. In other omics fields that are considered more mature, like genomics and transcriptomics, data sharing culture was very well established, especially since the Human Genome Project. The first version of the Human Genome was produced more than 20 years ago now. It is considered to be a good scientific practice, of course. We are lucky in the field because really we are really now, I think, in parallel to these other fields like genomics and transcriptomics, because the data sharing culture has really definitely consolidated in recent years. And this has been really a big change, and a change for the better. Scientific journals and funding agencies are two of the main drivers in this direction, but of course, I think that everyone in the field realizes that open science practices are more and more important, and therefore they have become more popular. So what is a proteomics publication in this year, in 2023? So as we all know, proteomics studies, like in any other kind of high throughput omics technique, generate potentially large amounts of data and results. So ideally, a proteomics publication needs to, first of all, summarize the results of the study, and second, provide supporting information for at least checking the reliability of any results that are reported. So therefore, I'm sure that you are well aware of this. The information included in a publication these days is included in the manuscript. This is what most people read. Then the supplementary material. This is basically what people that have a higher interest in some particular aspect of the work, a bit technical. Then people read that part as well, of course. And then what is becoming again invaluable and also important is to have the associated data submitted to a public repository so that everyone can access this data. And again, this has been triggered by scientific journals and funding agencies. So there are many journals that nowadays recommend and mandate, in most cases, submission to proteomics repositories. These are mainly the journals of the Nature group. They have been really pioneers in this context. Proteomics journals like MC for Proteomics, but also other journals like the PLOS journals or eLife and many others. And also what has happened also more and more in recent times is that the funding agencies are enforcing more and more public deposition of data to maximize the value of the funds provided. That means, of course, that the value of the funds doesn't stop at the end of the grant. It means that other people in the future can use the same data, can reuse the same data for getting new scientific knowledge. Also, proteomics repositories are very important for supporting reproducible research and reproducible science. So, of course, we should aim that what we are reporting in a manuscript, in a paper, is as reproducible as possible. Okay, so hopefully I convinced you why sharing proteomics data is a good thing and it is important. The second point in my talk is just to explain you how. For that, how you can do it. So for that, I'm going to explain about protein exchange resources and the PRIDE database in particular. Also, although I will be talking about other protein exchange resources. The short answer to this is that basically everything that is relevant for reporting an experiment. What is really mandatory to include in every submission is the original experimental MS data recorded by the mass spectrometer, the primary data, usually the raw data for sure, and in some cases the thick list. Then we have the process results that they include identification, but also quantification. Then what is really important, of course, is include as much experimental and technical metadata as possible, because of course the previous data types are not very valuable without the proper experimental or technical background. And then any other type of relevant information is also always welcome. For example, the search database or spectral libraries, or maybe potential scripts that were used. This is also important in the field because also the field needs to really develop in parallel to what is happening in other fields. So I don't know if you have heard the term, it's very trendy, fair, fair data, the fair data principles. And this is really a big development in biology. And the acronym FAIR means that the data needs to be findable, accessible, interoperable, and reusable. I'm not going to explain what all this means. I just wanted to say that you have heard FAIR, and at least I wanted you to know what this means. I'm going to be mentioning, of course, at the end, a big part of my talk is going to be devoted to the reusable part, but I just wanted to highlight that the development of open science practices and proteomics repositories are really important in the context of fair data in proteomics. Okay, so as I mentioned before, my team is responsible for PRIDE. PRIDE is the Proteomics Identification Database, and it's based at the EBI. It was started by my colleague Leonard Martens in 2004, 2005. And so it's now almost 20 years, 20 years old. And next year it will be 20 years old. So it's a lot of time. And really, PRIDE is the EBI resource that stores mass spectrometry-based proteomics data sets, including, as I mentioned before, the mass spectra, but also peptide and protein expression information, the results, any kind of related information. All mass spec proteomics approaches are supported, and really the amount of data in the public domain now is huge. By May 2023, there are more than 33,500 data sets in PRIDE. And this next slide is also to show you, how this data has been growing over the years. This is an histogram that shows the number of submitted data sets to PRIDE since 2010 to 2022. And this really shows the huge, this is not cumulative, this is year per year. So this is just to make you realize about how data sharing in the public domain has really grown dramatically in the last few years, and it's still growing. It seems that in the last few years is growing less rapidly in a way, and it's very nice that this happened. Of course, an average of 514 submitted data sets were submitted per month during 2022. And this comprises around 83% of all protein exchange data sets. That's why PRIDE is really the world leading resource in this context. And of course, this is thanks. This is no one knows this outside the team. So of course, Dipti knows very well all the work that is behind all this every single day. And because of course things need to work seamlessly for the external users, but really. This growth has been possible due to huge improvements in PRIDE infrastructure, including scalability, reliability, and automation. So it's also important, I think, to give an overview about which are the main organisms that are represented. So there is no surprise for you, I'm pretty sure, that the most represented organism is human, Homo sapiens, that comprises around 44% of the data sets, but this also includes cell lines. So yes, it's important to include that. And then, of course, the main mother organisms are just behind: mouse, Saccharomyces, Arabidopsis, rat, E. coli, Bos taurus, Drosophila, pig, C. elegans. So there's really no surprise here. But what is really important to highlight is that more than 3,600 taxonomy IDs are represented. So it's really, you are working in a particular organism, even if it's not really very widely studied. It would be strange that there is not proteomics data available in the public domain for this organism. So the workflow to submit data to PRIDE is, or to any other protein exchange resource, is that, of course, the data needs to be organized first. And that requires some work. And of course, there is always some learning curve. But I would say that over the years, the process has become more and more easy and straightforward. Otherwise, there wouldn't have been this kind of huge growth in the number of data sets in the public domain. So, the scientists submit the data using the pre-examination tool that I will explain in the next slide. And for that, again, we need the raw data, the results, and also any other kind of data. Then PRIDE curators check and process the data. And before, there used to be a lot of manual component here, and now it's more and more automated. and curators only look at the data if it's really required. And then, of course, the data gets submitted to Pride, and a data session number is generated. So the submissions to Pride happen mainly using what we call the Proton Exchange Submission Tool. It's a standalone tool that is developed in Java, and that was developed many years ago and is continuously improved. It streamlines the submission process. Again, the mandatory components are the raw files and the process results, and any other type of other files are optional. It captures the mappings between the files, it enables metadata notation, and then it enables the file transfer through the internet, to the ABI, which can happen using FTP, but also using Aspera, which is a technology that allows, in general, faster upload speeds. So this is kind of where you can access some of the documentation, and there is a manuscript that we wrote many years ago that summarizes the process. Of course, the process has changed slightly, but really not that much in the last years. So then, how to access this data? So this data can be accessed to the Pride web interface, the file FTP repository, or if you want to retrieve more data in a batch, you can do it using our application programming interface API, the REST web service. Some slide that I wanted to include here because it's important, because we usually get asked about this a lot, is that when does it happen, the public data release? So usually when submitters submit their data to Pride, but then also to any other Proton Exchange resource, the data remains private. And it's always private by default. We provide a username and password, and that can be used within the manuscript review process. So that can be provided to reviewers, that can be provided to journal editors, and the data is gonna be, it's always going to be private. But then, as soon as the publication gets published, the corresponding publication gets published, then the data should be made publicly available as soon as possible. So that means that in many cases, we will only find out if the submitter tells us that that's the case. Because, sometimes it's really not so easy to find out that a paper has been published and that it contains a data set that is reported in that paper. We can look for PhD identifiers in PubMed abstracts and also in full tests for those articles that are open access, but those are, the minority. So really, if your PhD identifier is not in an abstract or your paper is not open access, the paper may have been published, but the data will still be private because we will not be able to know that that's the case. So please let us know, because that will help to speed the process of making the data publicly available. So I have talked about Pride because, of course, it's the resource that I'm responsible for. But I'm also going to talk about other Proton Exchange resources. I think this is really important, and it's great that such a network has been developed over the years. And since I know that there are many Chinese students attending this webinar, I wanted to talk especially a little bit about IPROX, which stands for the Integrated Proteomics Resource. This is the URL. Is the Chinese proteomics resource, so you could call it the Chinese Pride. Although there's some things that are similar, but there are other things that are a little bit different. But again, the minimum common denominator is the same. The IPROX is focused on MSMS proteomics data. It's widely used by Chinese researchers, so I'm sure many in the webinar you are using regularly IPROX, because it's mainly used by Chinese researchers. And in the last few years has greatly improved infrastructure, including the addition of a reanalysis pipeline. So you can read all the details in these papers that are highlighted here. And in the last update that was published like 1.5 years ago, here is highlighted the improvements that have been happening in IPROX. So IPROX has now also programmatic access, API. It has new visualization features. It has also a reanalysis pipeline and other features that other proteomics repositories have, including others proteomics exchange resources. So it is really great that our colleagues in China and the people that are responsible for iProx, led by Junpin Zhu, are really keeping in parallel to the developments that we are doing in all parts of the world. We would also like to highlight Massive, which is the resource in the University of California, San Diego. Is led by my colleague Nuno Bandega. This is the URL. It's the largest proton exchange resource in the US. It also has kind of generalistic interest, focus on MSMS data. It's important to highlight that it contains really lots of data, including many data sets that were originally assigned to other PX resources, for instance, to Pride. There are many Pride data sets that are replicated in Massive. They have some tools available for users to analyze your own data, which is really, really nice. And they're also very actively reanalyzing data sets, as I will be mentioning later again. And one of the points that also they have been working very actively in the last few years is in the creation of spectral libraries, as I will also mention. So if you don't want to go to every single proton exchange resource individually, there is a website called Proton Central, which provides centralized access. It's a centralized portal for all public proton exchange datasets. So this portal is managed from Seattle. This is the URL. And basically what you can go there is basically to look for datasets in all proton exchange resources. So you could look there for datasets in Pride, in JPOS, in iProx, or in Massive. It needs to be public, of course, but because the communication between the individual resources and Proton Central doesn't happen till the corresponding data set has been made publicly available. So again, let me highlight the concept of the data publicly available for everyone in the community using any of the Proton Exchange resources there. And now I'm going to talk about the workflow. Maybe that's the most interesting part of my talk. I'm going to explain in detail some examples of data reuse for public datasets, because of course the whole purpose of storing that much data in the public domain is that that data is reused and is actively reused by anyone in the community. So this is basically the whole concept. People do an experiment. Maybe you do an experiment in the lab. Then you do your own interpretation. You generate new knowledge, which gets published, maybe in a paper. And then this knowledge is disseminated into a database. And then this knowledge from the paper and from the database maybe generate new ideas to do a new experiment, new interpretation, and this circle hopefully continues and continues without stopping. But now, because of all this data that is available in the public domain, it is possible to shorten this circle a little bit. So from the information that is available in databases like Proton Exchange resources of Pride in particular, it is possible to perform what we call in silico proteomics experiments that of course generate new interpretation and new knowledge that it can be published independently and then generate new ideas to perform new experiments, and of course to perpetuate this virtuous circle of proteomics data reuse. So the take-home message here, and I hope this is the most important thing that I would like you to take with you when you finish this webinar, is that it is not always needed to generate new data in the lab to get the conclusions or to do the studies that you need to perform. In fact, public data sets can be used in bioinformatics studies, and there are many options, and you will learn about some of them in the next few minutes. Also as complementary data to generated data in the lab. So it's important to highlight that it's not always required to generate new data. In some cases it's wiser to look to similar experiments in the public domain and to use that to confirm or to put them together with the data that you are generating. This is kind of the main message that I want to give. So in this part of the talk, as I mentioned before, I want to highlight the reusable aspect of public proteomics data. And again, this is really key in the context of FAIR and what it means, as I mentioned before. And also you want to keep yourself with an image of what I want to highlight here is that really the data in public proteomics repository is like the cave of wonders. So I remember many years ago when I saw Aladdin, and I remember this very well. And this is actually the case. Depending on what you want to do, the public proteomics repositories can really be a cave of wonders for you, because maybe you have there the data that you need, or maybe you have data there to text many of the ideas that you want to test. And it's really, really a cave of wonder. So in the next few minutes, I'm going to highlight some high-level examples of public data reuse. The classification that I have made is kind of, it could be made in other ways, but I think it will be useful to for you to highlight some of the uses that this data has. And the first use that maybe can be highlighted, and this is not in particular order, it's just an artificial order that I have created, is the creation of spectral libraries. So spectral libraries are collections of experimental spectra that that are accumulated, and that historically have been used for performing DDA experiments. So many of the proteomics resources, Pride, Massive, also Atlas, but also NIST, have provided spectral libraries for many years. This is also common in metabolomics, mass spec metabolomics. But and for that, the easiest way to do this is to put together a lot of data that has been made available in the public domain to create these spectral libraries. And also, spectral libraries are increasingly more and more important because, of course, as you probably know, they can be used for DI analysis. And one of the ways to perform this DI analysis is to use public spectral libraries for that. And this is why really there's now a renaissance of the use of spectral libraries in the field. In any case, if you generate your own DDA experimental library for a DA experiment, you should also share it in the public domain, because it is important for reproducibility purposes. The second example of use of public datasets is benchmarking of software, and this is really very common. And you will know about this if you are a bioinformatician or a computer scientist. You are well aware of this because it is quite common to do this. Basically, every time new software is published, then there is the need to perform a comparison with the previous tools, with the state of the art in the field. And this is done, of course, using mainly public data sets. And this happens all the time. There are, dozens of papers published describing new tools every week. And in many cases, in most cases, they use public data sets for the benchmarking. So in most cases, the raw data is used as the basis, but not always, because, for instance, in case of visualization tools or other type of tools, in some cases it is possible to use the process results that were originally deposited. So there are lots and lots of publications that make use of public data sets for benchmarking, and again, there are new tools every week. So this is just a very small sample of all manuscripts that use public data sets for benchmarking. So the third kind of type of data reuse, that is basically what I'm going to be talking about in the rest of this section, is data reanalysis. And data reanalysis, of course, it comprises to put together groups of raw files and to reanalyze them in different ways for different purposes. And really many things can be done in the context of data reanalysis. The first thing that I want to highlight is, for instance, to provide a consistent view on the proteome. So this has been done for many years by my colleagues in Atlas, led by Eric Deutsch or GPNDB, led by Ron Bibis. And what these resources have been doing in the context of identification data, they have been collecting raw data, and they have been reprocessing it using a consistent data analysis pipeline and an up-to-date protein sequence database. The advantage of this is that they can provide a standardized and updated view on the experimental data that is available, so providing a view on a particular proteome, of course, because one common analysis method is used. Of course, this has the inconvenience sometimes that the results can be different from the original author's view on the data. So the main resources that have been doing this for many years are DPNDB and PECAT-ATLAS, but also massively doing it in the context of the HPP project. And what is the HPP project? So the HPP project stands for the Human Protein Project, the Human Protein Project that was started by UPO in 2010 in Sydney, if I'm not mistaken. And it has been a tremendous effort to put together, to coordinate different groups of people coming from different countries, trying to detect all proteins in the human proteome. So it was the original objective for the project, and it has been now mostly achieved, I would say. If you see the last report of the HPP project, the report there, this paper is for the end of last year. They found evidence for more than for almost 18,500 proteins, so approximately 93% of all the proteins in the human proteome. So PECAT-ATLAS, but also Massive, have been reanalyzing a lot of data to provide this information to the HPP. But they have been mainly focusing on identification. I think it's important to highlight that there are other sources that they have been reanalyzing data with the view to provide a quantitative view on the proteome. And this has been done by the team led by Matthias Wilhelm, Proteomics DB in Munich. And this is really a very nice resource. And of course, they use a lot of data that have been generated locally by the group, but they also integrate data sets from other origins there. And they provide a very nice quantitative view on the human proteome. Another resource that maybe you have heard about, but I think they use a completely different method. I'm not going to get into the detail. Is PAXDB, the Protein Abundance Database. This is the URL, and the version 5 has been recently submitted to publication. It's in a preprint, and you can also acquire there a quantitative view on different proteins, not only human. And our contribution to this work has been in the context of a EBI resource that is called Expression Atlas, that historically has stored gene expression information coming from human, but also from the main modern organisms. And what we have tried to provide in recent years is to provide information about protein quantitative information there and integrate it to the gene expression information. So this has been our main objective. This is why we have been curating and reanalyzing data sets from PRIDE and integrating this information in Expression Atlas. And we have done this for a number of studies: cell lines and tumor tissue, human, mouse, and rat baseline studies. We have done also a pilot study with cell lines and serum, and all this has been published recently. So far, approximately 100 quantitative proteomics datasets are integrated in Expression Atlas, and this is really how it looks. But without talking about more about the details about this, the most important concept here is that groups of raw files coming from different data sets can be reanalyzed together to provide either a qualitative or quantitative view on the proteome, and this is what an increasing number of resources are doing. Another type of a study is what we could call meta-analysis study. Again, what I've been providing so far is kind of a meta-analysis, but of course there are different types of meta-analysis. Here, the idea is to put data coming from a lot of experiments together to extract new knowledge. So, for instance, data integration of experiments done in different labs at different time points. And for this approach, data is mainly reanalyzed, because that's considered to be the best way to make the data more comparable. So again, there have been lots of studies. I mean, what the proteomics repositories are doing, it could be also considered a meta-analysis, but I thought that deserves their own category. But there are many studies that are performing this, are putting different data sets together with different objectives, and trying to extract new information by putting all that information together. So there are several papers, for instance, in the context of protein complexes and protein-protein interactions. Also some papers in the context of clustering of MSN spectra, and many others. And this is kind of something that has happened also in other fields. So there, if you go to investigate what has been done in transcriptomics, there have been really a lot of studies, meta-analysis studies, where a lot of information coming from a big number of transcriptomics studies have been put together. Okay, so another kind of subtype of data analysis studies, again, different purposes, there are different ways to do this classification. It could be in the context of artificial intelligence, machine learning, or deep learning approaches. As I'm sure you are aware, these approaches are more and more important in biology and in proteomics in particular. There is a very nice review paper by the Matthias Mann's group that was published almost a couple of years ago, and you haven't read it and you are interested in this topic, I would invite you to read it, because it contains this nice figure where it summarizes the different efforts that have been done in the field to improve the proteomics experimental workflow using machine learning approaches or artificial intelligence approaches. So as you can see, this figure includes all the steps in the proteomics analytical workflow, going from the sample, then it's the digestion, the liquid chromatography, the ion mobility. Then there's one aspect, the vector fragmentation. Then there's two spectra, then the vector and protein identification, and the protein quantification. And there have been many groups that have contributed with new tools to improve these different aspects in the proteomics. So there has been really a revolution in this context. For instance, in the context of prediction of peptide fragmentation and rescoring, and many other steps. These are just some of the tools that are there. I would like to highlight that public data sets are very often reused as training sets, and also and this is happening more and more, of course, because the more data, the more reliable the predictions will become. There are many papers that have been published in this context in recent years, and again, these are just a small subset. And what is clear, at least to me, is that these approaches are only going to become more and more important in the near future. And for that, it's also key that there is more and more data in the public domain. Okay, and just to finish this part, I would like to highlight what I call another type of data analysis, what I call new analysis perspectives, or repurposing. That's another way to call it, where well, you know, the data is being analyzed with a different purpose to the one that was intended in the original study. And there are two main examples of this. One is the proteomics approaches, and one is also the new PTMs. So in the context of proteomics approaches, just a definition, in proteomics approaches, proteomics data is combined with genomics and transcriptomics information, typically by using sequence databases generated for DNA sequencing efforts, RNA-seq experiments, RABO-seq approaches, or London calling RNAs. So proteomics has been historically used for genome annotation purposes. And there have been a lot of studies where MS data, proteomics that has been reused for this purpose. So again, no new data was generated. The data was taken from public repositories, and there have been really many publications where this has been achieved. So discovery of short operating frames, translating London calling RNAs, etc. And this has happened mainly for human, but also for other model organisms that I think I didn't include the slide at the end. And just to finish this section, I also wanted to highlight that new post-translational modifications have been detected, again, by reanalyzing raw data with new hypotheses in mind, not taking into account by the original authors. There are some examples of publications from a few years ago where they were using phospho-enriched proteomics data sets, and these are some of the modifications that were found. But for instance, there's a very recent example from a Chinese research group that was published last year in Nature Methods, where they reported the widespread lactylation in the human proteome. And for that, they use different public data sets, and also they use a spectral library. Okay, so I'm finalizing, but I thought that it was good to highlight that, of course, there are some bottlenecks for data reuse of public proteomics data. for the people that are outside the field, really, proteomics can be quite complex. So that means that there is a steep learning curve for researchers working in other fields, transcriptomics or genomics, for instance. Mass spectrometry raw data is big, and that requires an infrastructure. Then, in many cases, it's a high performance computing infrastructure. And it is a problem that Windows is often a requirement for analysis software in proteomics, although this is changing very rapidly. Software analysis methods are, in some cases, very dynamic. So that also adds a difficulty. And of course, another thing that I would like to explain a little bit more in detail is that in many cases, there's a lack of enough metadata annotations in public data sets. So there is the need to spend quite some time in curating the data sets. And this is because of historical reasons. So when we started Protein Exchange, the main focus was put on providing or generalizing the availability of public data in the field, which was not common in the past. So then the focus was put in generating a data set, general description, and then in providing the data files. So what we call again the raw MS instrument files or the result files containing identification and quantification. So really what is missing, what has been missing for many years is the mapping between the sample characteristics and the data files. So this is what is summarized here. The structure metadata to link individual files to sample characteristics is mostly missing. Very often some information can be inferred, for instance, based on file names, but this is far from ideal, of course. Structure metadata to link to individual samples makes it hard to annotate and reuse the data. So also this would be very important for linking samples in multi-omics studies. So in a work that was led by the Seth from my team, he led the development of a format that is called SDRF proteomics that is actually a new flavor of a format that already existed in the transcriptomics world that was developed originally by our colleagues in Array Express and Expression Atlas in the ABI. And what this format provides is basically that. It provides the link between the sample and the data files. We already have the dataset general description. This is already available for every Protein Exchange dataset, but the information for linking the samples to the different files and the experimental design is what is provided in this format. It's a tab-delimited file, and we are promoting it as much as possible. And its adoption is kind of increasing in the field. So you want to know more about the format, is this GitHub page. And the way to create the files at the moment is mostly manual, so using Excel or any kind of spreadsheet software. But there are now starting to be tools that can be used to create SDRF proteomics files. So this is the work of Tim Eccles from the Compomics group in Ghent. So he has developed a tool that can that is a first attempt to generate SDRF proteomics files in a user-friendly manner. This is the URL where you can access that tool, and hopefully this and other tools in the future will make SDRF and the annotation of data sets more comprehensive. So I'm just finalizing now. I want to highlight that, for you to realize that data in Pride is really reused more and more. In the last 12 months, more than 1.1 petabytes of data have been downloaded by more than 180,000 total unique hosts, and many of them from China. So that's from really China is our most most popular place in terms of data downloads and data reuse, at least from that perspective. And also, Pride datasets have been reused in more than 550 publications. We are tracking this. Of course, this is an underestimation because this only includes the ones that we can really track properly. Our vision for the near future is that data from Pride and other protein resources needs to be integrated more and more in other biological resources. Here, we include in resources in the BI, like Expression Atlas, UniProt, PDBE, Protein Data Bank, or Ensemble, because really, and I think this is important for the field as a whole, proteomics data needs to be made more accessible to people that are not experts in the field. So people that are not experts in proteomics. Good. So just the last part of my talk, just one slide to give you some pointers for future perspectives. So Data sets, in my view, are becoming larger and larger, of course, with larger cohort studies and maybe single-cell proteomics in the future. So it means that for proteomics resources, there will be the need to have continuous improvements in infrastructure to be able to deal with these larger and larger data sets, and to also make some missions feasible for you, for the scientists. Then we need to continue enabling improvements in dataset metadata provision. There is also the concept of fair data that I hope you learned if you didn't know about it before. And it's really key for the field to integrate proteomics data in other databases. There is some kind of new data types that are becoming more and more popular that are not non-MS based techniques like Olink and somalogic assays, and we will need to store those and make them accessible. And yeah, any other topic you would like to raise in the Q&A session, I will be happy to follow. I just have to acknowledge everyone in the team, led by also Jacet, who is leading many activities in my team. And also, I would also like to acknowledge the work of my colleagues in other protein exchange resources. Of course, Dicty, especially because he's responsible of many of the data submissions that are made to Pride, and her work is invaluable in this concept. If you want to know a little bit more, there are several publications that you can look at about Pride, protein exchange, iProx, and others. And that's it. I will be happy to take any questions. I think that it took me like 5 minutes more than what I expected, but it always takes a little bit longer, I think. So I'm happy to take any questions.
Yeah. Thank you, Johan. It's like I know there are a lot more, and it's very difficult to capture in such a short time frame. But I think the participants and everyone have got at least a nice overview of what we do, what Pride is, and how the importance of public repositories and how and where the data has been reused. So just not to take more time here, I would start with the questions. I will keep some questions from the panelists from at the last, but I will ask from the participants first. So the first question is, how do you envision the future of data repository, knowing that data sets nowadays are containing more and more samples, for example, single cell, and some instruments are giving very large raw data?
Yeah, so this is what I explained in my last slide. So this is a continuous struggle for any proteomics repository in terms of infrastructure. So of course, that usually happens. You only realize that things don't work, or you only kind of realize that Pride is there if things don't work in a way, because then it was when you realize, oh, this is something is happening there. That's usually what happens to us. So it's really, it means that on top of everyday work that we need to do, we really need to be continuously innovating in terms of infrastructure and in being able to manage increasingly large amounts of data. We are lucky in a way because we are in the EBI, as I mentioned before, and we have the experience that our colleagues in genomics, for instance, have been experiencing for many years. They still deal with much larger data sets that we are doing. And then for us is, the first thing to do in many cases is to ask them about, how they manage this, or how they thought about these problems before, because we are lucky from that perspective. Maybe in other places it's not possible, but for us is possible to do that. But yes, it's a struggle, and it is kind of unavoidable. So it's clear that larger and larger data sets will be made available all the time. And in terms of storage, and this is another aspect, at least at the moment in the EBI, proteomics is still a tiny fraction when compared to, again, large genomics sequencing projects. So from that perspective, we are kind of, safe. But it's true that maybe at some point, there will be problems because of this. So, yeah, so I don't know if I could reply.
I think that's
can I have a follow-up question?
Yeah.
So compared to the genomics data, you have the proteome data deposition increasing over time. You have a very steep increase. Do you see the same trend for genomics in the past, let's say, 10 to 20 years? How do you see the future of proteomics? Do you see the future the proteomic data will someday exceed the level of genomics, or you think it's not in the same scale?
There are two different points here, Jonan. So in genomics, it's true that there are many data sets because there are very large sequencing centers in the world that use genomics, so they do it very well and they generate large amounts of data. So the profile of submitters to, imagine, for instance, the resource of the ABI, the ENA, which is the kind of GenBank for sequencing data in the ABI, or the EGA, which is the kind of control access protected version. So they get increasing sizes in terms of data sets, but most of that data sets come from, a relatively small number of places. I think what is different in proteomics is, you have this, or at least at the moment, is that data is generated, by thousands of labs around the world. And this is kind of a different kind of a scenario. That's what happens in genomics, at least from my perspective, where most of the data are produced, in a smaller number of centers. So of course the data is larger and larger because the throughput is larger and larger for genomics. I think it is difficult to envision, at least as of today, that the amount of proteomics will kind of, be larger than in genomics, because of course there will always be a lot of things to sequence, organisms or individuals, or I don't know. But I think at least at present it's difficult to envision that, that the volume of proteomics data will kind of overtake the sequencing data that is being produced. I think it's important to highlight that again there are different profiles again that what is happening. Yeah.
Okay, so I hope that answers the first questions. And now the second question is, availability of sample metadata is crucial for identifying samples, which are important aspects of open data practices. Single large number of data in PRIDE have metadata missing in them. So what are your suggestions for data submitters to PRIDE? Maybe this could be the takeaway message for the people who are looking forward to submit to the PRIDE. So yeah, please, Leon, go ahead.
So as I tried to explain in the last few slides, so again I went a little bit faster. So again, proteomics change is really not at all. We started from a little more than 10 years ago. And at the time, the perception was that what we needed to achieve was to get data availability in the public domain, that everyone would be doing this. So with that in mind, the opinion from different people that were involved at the time was that the priority should be put in that, and that the priority would be moved to provide more and more data interactively. Okay, so this is what we had in mind when we started this. And it is clear now that in many cases, the lack of metadata is really an impediment for data reuse. So there are different aspects to this. On one hand, so I'm just going to highlight some of the problems and some of the possible solutions. So in the last couple of years, again, in the work that was led by Jacet and others in EUBIC and PSI, there has been the development of this format that can enable the annotation of data sets in this manner. But, this format has to be generated. At the moment, most people generate it manually because the adoption is still relatively low, but I have to say that it's kind of growing. So ideally, in the future, we would have tools, I don't know, like Mascon or Mascot or others, in the DIA world, that would export this information. And then in that format will be available for you to upload into PRIDE or other resources. But also historically, the field, many of these tools that are used for analysis, they don't require basically any kind of sample related information when you are performing the analysis. So it is kind of it's a problem that it will be resolved, but it will be resolved in in a iterative manner and in kind of a slow manner, I think, in a iterative manner. So what is possible to do right now, if you want to submit to PRIDE, is to create these SDRF files, proteomics files manually. there is this tool that I introduced at the very end that has been now just developed by Tiner, that at least for datasets that are not very large, it works well. But of course, this is just the first version. Will need to be made better in time for sure, like it happens with any tool. So so I think that the future is bright, just to be positive. As you also saw, we get more than 500 datasets per month. In Pride, we are not so many people, and we don't have many resources to do this. So this, what I want to highlight, this is not a work that is, for Pride only or for Massive or for, it's a work for the community as a whole. And this is a problem that we need to solve all together. It is unrealistic to ask, that one team solves all these problems just at once, because it's really impossible. So the situation is much better than what it was 2 years ago. The situation will be better in 2 years. Let's put it this way. But I hope that I think that at least now there is a mechanism to do it, and there are also many data sets that are being annotated a posteriori, and the SDRF files are being made available. But yes, this is an interactive problem that will be solved in time.
Yeah, so okay, so the last question from the participant is many journals have dedicated data availability section where
Yeah.
accession IDs are mentioned. Have you considered using these to detect publication with Pride IDs?
So of course we do that. So I maybe I didn't explain myself very well. So we do that. We check the abstracts of the publications, and we check also the full test of publications. But we can only do that if the publication is open access, and also if it's fully open access, because there are different types of open access, and then you can only, well, I'm not going to get into the details. But if you have, submitted your publication, and the publication is not open access, so it's under a paywall, then it's impossible for us it's impossible for us to know automatically whether the data set has been published or not. It's just we don't have access to that. We can only have access manually, one person going paper by paper, which is not feasible. I'm sure you understand. So it is not the problem of the data availability statement. That's of course there, and we can look for it. Is that that in order to access that, the article needs to be open access, and most articles are not open access. So, or at least a majority of them are not open access. So I hope I could answer the question.
Yeah. And there is one question, I have answered it, but if you want like to add something on it, is like, are there any option in Pride to keep raw data private even after publication? These options exist in Massive. So yeah, if you want to add anything to this, I have answered that yes, the raw data are public, but if you want to add anything to this.
So, I mean, the policy for proteomics change is that as soon as the publication is out, the data needs to be made publicly available. So, and again, we don't even check this manually. There is a pipeline that is launched, and then it looks, and then it is kind of a policy, and it shouldn't be broken unless there is a very good reason for that. Can there be good reasons for it? So for me, there should be no justification if you are using data in two different papers and the two of them are not published. So we get this query every once in a while. As soon as one of them is published, then I don't see why the data shouldn't be made publicly available. And then there is another reason that is a little bit more, and I didn't get into the details in this talk because it was just too much. But there is the issue of data that can be considered to be sensitive for legal reasons. Data coming from human cohorts and human. For that, in genomics and transcriptomics, there is what they are called control access repositories. So we don't have yet that in proteomics. We're actually working on that already. But in some cases, kind of a proxy to kind of simulate this can be to keep the data private forever. We don't do this in Pride because we cannot do it. But I know that in some colleagues have used this kind of method or kind of a shortcut to try to deal with this situation. But that's kind of a hack, let's put it this way, and this is not sustainable in the medium long term. In the medium long term, what is sustainable here is to develop a control access infrastructure, so that for this sensitive data, so that people will need to ask for access to this data before they can access it. So I hope I was clear.
Yes. So there are just two more questions from Tianan, and
Maybe I can ask a question.
Okay.
Since now this is a writing course for people in this field, you mentioned some data more can be reused. You showed some example. Do you have any suggestion what type of data sets are more frequently reused by others? So what should we? What are good practice for when writing this data set could be potentially frequently used?
So the data that are usually more reused are the ones that are, there are two aspects to this. One of them are the ones that are more scientifically relevant. So, the ones that may in some cases get generated by, a famous group, and they're published in high-profile journals. So there are some of the data sets that get more reused. So that's one aspect of that. The other aspect of that, of course, the data sets that get more reused are the ones that are annotated better. So the ones that, people don't have to really do a lot of work in order to, again, find the relationship between the samples and the files. So I think those are the two groups of data sets that are reused the most. In the context of machine learning and artificial intelligence, there's really not, there are a lot of technical data sets that are reused a lot because they are very useful for the technical objectives of the, of the machine learning kind of methodology. So from one hand, yes, it's true that, the data sets that, that are kind of become more relevant get reused more, but actually many data sets get reused. And some of them are kind of, relatively small technical studies, and it's because of all these possible applications that there are.
Okay, so another question. Any comment to the difference of annotating data files for TMT, DIA, and SM and PRM? The TMT is normally they have several fractions, and those fractions are labeled by sometimes 6 plex, some 10, some 16. But DIA mostly single shoots, and for SM and PRM they also mostly single shoots. So do you have any advice?
You mean for you asked at the beginning the annotation of the file, no?
Yeah.
So of course, everything that's if there is one sample per MS run, it is much easier, and if there is no fractionation, then it is much more easier to annotate than if there are different channels in, for instance, in a TMT experiment. So the SDRF proteomics file format supports all those scenarios, supports fractionation, supports channels. But I wouldn't say that it's more easy or more difficult. It's just that it's more complex because, of course, in a TMT MS1, you can have, as you say, maybe more than 1,000 different samples, and they need to be annotated differently, and they all need to be mapped to the corresponding channel and to the corresponding file. That's a little bit more difficult than just one sample in one MS1. That's what you can get, for instance, from a DIA experiment. At least in most DIA experiments so far, I know that there is now starting to be leveling there as well. So, but it's not because it's more difficult conceptually. It's because it's more difficult because there are more samples involved. I don't know if I explain myself correctly there.
I can just add, if I may, here that Tianan, you can find annotated projects or the samples in the private GitHub page. I can share with you there, and the documentation is all very well explained, and you can always ask private support, or you can write to us. So you can definitely have a look in our GitHub page for sample annotated data sets, or any example files are there to see examples for TMT or DIA data sets, how they are annotated. So you can always, you're more than welcome to look there.
As I said, it's also good, again, this tool that, again, is very new. So, of course, it's far from perfect at the moment, but the tool that has been developed by Compomics Lennart Martenstein and Tine in particular, is also a good attempt to a good first attempt to try to facilitate this process in a kind of more user-friendly manner than just going through individual Excel files.
So just now, I noticed you have provided some online tools to reanalyze the data from Pride. I know from genomics field, so their data are more, the data standards probably more universe. So there are a lot of third-party tools to analyze genomic data. But currently for the Pride online tools, are they all provided by you? Your small team actually did amazing work. Only about 10 people. But do you open to other third-party collaborators who provide more tools to reanalyze the data?
So I mean, so the different answers to this. So first of all the reanalysis that we do in the team, we just do it ourselves or with very close collaborators, mainly because of grants. No, so that's kind of similar kind of people that share common interests. So that's kind of different. I think that what you are referring to is that it would be ideal, and I agree, it would be ideal to have an ecosystem of tools that would be where all the data, all the Pride data would be available. At the moment, you know, especially if you are in China, it's a huge difference. I cannot even imagine. You need to download the data from Pride, which can take, you know, I don't know, ages. I don't know how long it must take. We were talking about this at some point. That's also why it's good to have iProx in China, because of course, apart from it's because the downloads are made more manageable. So ideally, it would be ideal to have, you know, what is called the concept, the tools closer to the data. That means that could be like an instance that would contain, I mean, at least some of the high-profile Pride data sets, and the tools would be located there. So, you know, external users from China, from, you know, from wherever, we could be able to analyze public data there. Maybe, you know, some kind of cloud-enabled infrastructure or something like that. So that would be incredibly nice to have, but it is not. It is difficult to make it feasible. So of course this costs money, and there have been kind of initial ideas around that, but I think it would be unfeasible in the medium long term unless, you know, there is kind of a constant or, you know, a reliable source of funding that, you know, some projects that we could kind of, you know, use in order to do that. Because what people don't, or maybe don't realize, is that really computation and all these kind of cloud infrastructure, they are really expensive to maintain, especially for huge amounts of data. So this would be ideal. It's a very nice vision to have, but at the moment, at least for proteomics and at least for, it's really difficult to materialize because of, you know, the practicalities of, you know, or where to get the funding to get this kind of infrastructure running. So at least from my perspective.
Thanks.
Okay, so if there are no more questions from anyone, I would like to thank you, Juan Antonio, for his very enlightening talk. And I ask all the participants again to look for like our next announcement for the ETC auditorium. And I think we can finish. And thank you everyone for attending today's seminar. Okay.
Yeah, thank you very much.
Thank you.
Thank you. Bye bye.